Welcome to This Explainer. Today, we're turning some pretty complex bioinformatics into a full-blown 3D scientific investigation of your gut's hidden superpowers. We're going to take a journey from the very large, the human digestive system, and zoom all the way down to the incredibly small. We are literally diving right into the genetic code and the 3D structure of a single, absolutely fascinating bacterial protein. So, think about this for a second. Where do your essential vitamins actually come from? You might think it's just your diet, the fruits, veggies, maybe the supplements you take. But what if I told you there are literally microscopic factories inside you, working overtime right now to produce these vital nutrients? Turns out, we've got this incredibly profound symbiotic relationship with the microbes living right inside of us. Let's quickly map out our investigation today. We'll kick things off with the gut's vitamin factory, then meet our specific probiotic strain, KCA1. After that, we'll examine its folate-producing engine called DHFR, uncover its unique genetic fingerprint, build a 3D protein model, and finally wrap up with why this discovery really matters for human health. Section 1. The gut's vitamin factory. Let's start with the big picture of how the microbiota in our colon actually produce these essential vitamins for us. You see, our gut microbiome is absolutely packed with genetic info capable of synthesizing water-soluble B vitamins. We're talking biotin, folates, riboflavin, thiamine, and cobalamin. And unlike dietary vitamins, which get absorbed higher up in the small intestine, these microbial vitamins are produced and absorbed right in our colon. Basically, the colon acts as this massive repository for these nutrients, which directly impacts our overall systemic vitamin levels. Today, we're zeroing in on one of those in particular. Folate. Folate is absolutely essential. It regulates the amount of tetrahydrofolate in our cells, which basically makes it the fundamental fuel for DNA replication and build-in healthy blood cells. Because it's so vital for cell growth, figuring out exactly where we get it from is a huge deal. Section 2, meet probiotic strain KCA1. Let's go ahead and introduce our prime suspect in this microscopic mystery, Lactobacillus pentosus KCA1. Now, this isn't just your run-of-the-mill bacteria. The KCA1 strain was specifically isolated from a healthy Nigerian subject. And here's where it gets really fascinating. Previous physiological studies actually showed that when researchers fed this specific strain to Sprague-Dawley rats, it enhanced their hematological parameters. Simply put, it noticeably improved their red blood cell health. And that biological clue leads us right to our central question for today's explainer. What exactly is inside KCA1 that gives it this kind of blood-boosting superpower? How is this tiny microscopic organism enhancing its host's blood health from deep inside the gut? Section 3. The folate engine, DHFR. To find the answer, we need to transition from basic biology and dive into the incredibly powerful world of bioinformatics to look closely at the genome. By using bioinformatics and some really complex computational tools, scientists analyzed the entire chromosome of the KCA1 strain, and they discovered something remarkable. It has all the complete genes, the open reading frames, required for de novo folate biosynthesis. It's essentially a fully equipped microscopic vitamin factory. Now, looking at this intricate metabolic map generated by the RAST subsystem, you'll notice the green boxes labeled DHFR. That stands for Dihydrofolate Reductase, and it's the main engine of folate production. Here's the kicker. While some related bacteria only have one gene for this, the RAST system showed that KCA1 is highly equipped with 3 distinct genes for DHFR, so it is heavily invested in pumping out folate. Section 4. A unique genetic fingerprint. Having the engine is one thing, right? But what makes KCA1's specific DHFR engine completely different from the ones found in its closely related bacterial cousins? To figure that out, researchers brought in an algorithm called ClustalW. You can think of this like a superpowered spell checker for genetics. They took the amino acid sequence of KICA1's DHFR and lined it up side by side against 16 other lactobacillus strains from protein databases. Basically, they were hunting for structural differences, spotting the typos in the genetic code, if you will. Well, that alignment revealed a highly unique genetic fingerprint. Right at position 85 in the amino acid chain, KCA1 has a unique substitution. It has the amino acid alanine. Meanwhile, almost all of those bacterial cousins have aspartic acid or threonine at that exact same spot. It's just a tiny single substitution, but it acts as a massive distinguishing marker, proving KCA1 is genetically distinct. Section 5. Building a 3D protein model. Having locked on to this unique genetic sequence, the next step is visualizing it. Researchers used an advanced automated platform called I-TASSER to generate a 3D biological mugshot of this exact protein. Now, I-TASSER doesn't just guess. It uses complex physics and known structural templates to predict exactly how that linear chain of amino acids folds up into a 3D shape. For the KCA1 DHFR enzyme, it predicted a highly specific structure: 5 alpha helices, 8 beta strands, and 12 potential active binding site residues. That's where the actual chemical reactions take place. Just look at this beautiful 3D ribbon structure generated by the modeling program. The alpha helices are highlighted in red, and the beta-pleated sheets are in yellow. This visually proves what the KCA1 DHFR enzyme actually looks like out there in three-dimensional space, finally giving researchers a physical map of the enzyme's active sites. Of course, in science, you always have to validate your models. When they mathematically compared their new KCA1 model against a massive library of known proteins in the protein databank, the results were striking. The top hit was a protein called 1ZDRA, with a TM score of 0.971. In the bioinformatics world, a score that close to 1 means extreme confidence. It confirms, without a doubt, that this model perfectly matches the functional profile for a real folate-producing enzyme. And these comparative structures brilliantly illustrate that validation. You're looking at a 100% van der Waals forces overlap. The new KCA1 model sits neatly alongside established, real-world template proteins like 1ZDRA and 2ZZA. The shape, the binding sites, the structural folds, they all align perfectly. It proves the computer wasn't just hallucinating. It mapped a functionally viable, real-world biological engine. Section 6. Why this discovery matters. So what's the ultimate verdict of our scientific investigation into this tiny microbe? Why did we just spend time mapping out the 3D structure of a single bacterial protein? Well, the researchers actually summarized it best in their conclusion. This unique 3D structure is way more than just a neat computer graphic. Because of that unique alanine substitution, it represents a brand new template for structural genomics. It's a physical model that scientists around the globe can now download and use for computer-aided drug screening. So the crucial takeaway here is that this bioinformatics deep dive accomplished 3 major things. First, that unique genetic sequence helps us definitively distinguish KCA1 from other probiotics out there. Second, it perfectly explains the biological mechanism behind why feeding this bacteria to rats enhanced their blood health. It's a massive folate producer. And third, it gives the scientific community a literal 3D blueprint to screen new drugs and better understand how to boost human health from the inside out. It's honestly mind-blowing how much power is packed into a single microscopic organism living right now in our digestive tracts. I'm going to leave you with this final thought to ponder. If one unique genetic sequence, a single amino acid substitution in a single strain of bacteria, can actually enhance our overall blood health, what other microscopic miracles are hiding right now in the depths of our microbiome's DNA? Thanks for joining me on this explainer, and keep that curiosity going.